Now, this question is on investigation. Please, one important thing to always take note of is the aim of the investigation, and that will give us an idea of what the question is about. This says the student investigated the effect of cooking time on the vitamin C content of water. So let's come to the main information. It says cooking fruit and vegetable water reduces their vitamin C content. Vitamin C is transferred from the fruit and vegetable into the water by diffusion, meaning the more you cook the fruit and vegetable in water, more vitamin C diffuses out of it. All right. Now it says DCPIP, of course, is a reagent to test for vitamin C, and usually it turns colorless. It becomes colorless because the original color of DCPIP is blue. Now let's come to the steps the students adopted in carrying out this experiment. It says a piece of foil was wrapped around the beaker. All right. The base and sides of the beaker were all covered. Now the top of the beaker wasn't covered. Step 2, a piece of citrus fruit was put into the beaker. Step 3, hot water was added to the beaker so that the piece of citrus fruit was covered. A stop clock was started, meaning the time for the experiment. So we have the citrus fruit in the beaker along with the hot water. Now it says 1 cm cube sample of the liquid in the beaker were taken every 1 minute for 5 minutes, meaning the whole duration of the experiment took 5 minutes. At each minute, a cm cube of the liquid in the beaker where we have the citrus fruit and hot water was put into a separate test tube. A 5 cm cube syringe was filled with DCPIP solution, and any air bubble present in the syringe was removed. 1 drop of the DCPIP solution was added to the test tube containing the first sample. Now that's a procedure for getting our result. Now the test tube was shaken 5 seconds to mix the content. The color of the sample in the test tube was observed. Now the student repeated step 7 until the sample remained blue. Meaning there was no more vitamin C in the sample. Now the solution of DCPIP the volume of DCPIP remaining in the syringe was recorded. So what was measured? The volume. How do we take a result? The volume of DCPIP remaining in the syringe was recorded. So those are the things the students recorded. Now step 9 says the syringe was refilled with 5 cm cube of DCPIP solution. Then the steps were repeated for the 2nd minute, the 3rd minute, the 4th minute, and the 5th minute. Since we have 5 first minute, 2nd, so that was what was done by the student. Now let's see. It says figure 1.1 shows the student's notebook where they recorded their results and calculated values. So in the first minute, the 1 cm cube sample taken, okay, the volume of DCPIP remaining in the syringe was 3.6, meaning that they used 1.4 cm cube of DCPIP in the first minute. In the 2nd minute, they used 2.2, 3rd minute, 4th minute. For the 5th minute, of course, we have to find this out. So let's go to the first question. It says, using the equation, calculate the volume of DCPIP added to the sample taken at 5 minutes. Now, that's the formula. Starting volume, of course, we know is 5 cm cube. What is the volume remaining? From the table, the volume remaining is 1.6. So we can just subtract that from what we have, and we know the volume that has been used, 1.6 cm cube. Of course, that's what 3.4. You don't need to repeat the answer. So your answer is supposed to be 3.4 cm cube. Every time we calculate, we must always indicate unit where necessary. Very important to get maximum mark because the unit has been indicated here. We don't need to repeat that. So that's 3.4. So that's the answer to the first question. Now the question says prepare a table and record the results shown in Fig 1.1. There are important things to note when preparing a table. Your table must have a header, okay, with unit. Header with unit. Then in your table, you must also have the rows or columns, okay, closed. Okay, what do I mean by that? You close all borders, you close your borders, all right, and then you use appropriate 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 decimal places where necessary, or follow an appropriate pattern. All right, let's come back to the table. Now it says volume of DCPIP remaining in the syringe, volume added. So what we need is the volume of DCPIP used, okay, volume used, and of course the numbers of times we did the sample. So of course you have to draw this with a ruler, okay, you have to use a ruler to do this, but because I am using my pen, I don't have to have a ruler. So let's have all borders must be closed. So you have the sample, okay, sample of citrus fruit, okay, and then you have volume of DCP IP used or added, okay, and you put the unit in cm cube. So you have first minute sample, okay, second minute, third minute, fourth minute, and of course the fifth minute sample. And you close your borders like that. You close your borders, close your borders. Of course you do this with a ruler. You close your borders, and then you fill in the values. Remember, you have to follow the line of answers. This is in one decimal place. So we have 1.4222832. So we have 1.4, 1.4, 2222832, 1.4, 2.2, 2.8, 3.2, and the answer we calculated out is 3.4, all to one decimal place. Now you don't need to repeat cm cube here because you've already added it to the header, okay, so that's why your table requires a heading. So take note of this important things when making a table, okay. So you close all borders, all right, you use your table as shown. That's quick 3 marks for you, and then we move on to the next question. The next question is state the conclusion for the result. Like I told you, every conclusion must go back to the aim of the experiment or the investigation. Now, what is the aim of the investigation? Effect of cooking time on vitamin C content of water. Of course, the vitamin C content of water would increase by diffusion. So what do we see in the result? As the cooking time increases, okay, the diffusion of vitamin C into the water increased. So that's what we have. So you say as cooking time increases, okay, the diffusion of vitamin C into the water increased, okay. So that's it. So you can put some the cooking time of the citrus fruit increase. So what, that's just good enough. So you can use any other word, but the details must be well and clearly stated. So that's that with that. Now this
This is what we do in the masterclass, really. We try as much as possible to ensure that we're able to properly, right, teach learners how they can properly understand, interpret the question as much as possible. We dive deeper than just a past paper to ensure that at the end of the day, they're able to fully understand any question that is being administered to them.
state the independent variable in this investigation. Independent variable means the variable that the experimenter manipulated. So what was manipulated? We manipulate the time. Okay, we manipulate the time of the what? The citrus fruit in water. The time the citrus fruit stayed in water. The time of the citrus fruit in water. That was what was manipulated, because you do the first minute, second minute, third minute, and fourth minute. That's what was what manipulated. Okay, that's what the experimenter changed. What was changed? Okay, change, variable change during the experiment. All right. Now state the purpose of wrapping the beaker in foil in step one. Why did we wrap the beaker in foil in step one? Now in step one they said, because we are trying to deal with temperature and diffusion, we know temperature is a factor that affects the rate of diffusion. So we have to maintain the temperature to keep the water as hot as possible. So it's to, okay, for insulation. Okay, for insulation. Other words you can use is that to what? Maintain, to maintain the temperature. Okay, any of this will work. You could also say to reduce heat loss. All right, that's that for that. It's 1 mark, so 1 point is good enough. Now explain why. Explain why it is important to remove the air bubbles from the syringe in step 6. When the volume of DCPIP was measured with the syringe, we needed 5 cm cube. If bubbles stay in the syringe, of course, it's going to affect the original volume of the DCPIP, and that will cause an inaccuracy in our result. So we say we need to remove the air bubbles to obtain accurate volume measurement, okay, of DCPIP. Okay, so our result, so the result is adequate. Okay, so that's that with that. Let's go on to the, so that's all with question 1, and very easy. Just follow the line of process and answer what you're being asked. We are done with question 1. Let's go to question 2, and it's on planning and investigation. The important thing about planning and investigation is that you have your own decision to take in how you plan the investigation, but you must follow the rule of carrying out an investigation. And total marks usually is 6 marks, so you don't need to write too much and take all your time solving. If you remember the paper 6, the entire paper is just for an hour, and you have about 3 or 4 questions to answer. So you have to be careful and time conscious when planning your investigation. You have the laid down or rules to answering, or things or guidelines to answering such questions. You have to state your independent variable. You have to state the variable or your method. Okay, then you state your constant variable. All right, you state your dependent variable you're going to measure, the dependent variable. You also tell us how you will measure it, your result, and the instrument you are using to take your result, to measure your result. Then you give us the reliability, okay, how you make your result reliable, and you talk about your safety precaution or safety measure. So these are the essential things to take note of when doing paper 6. So let's go through this. This one says plan an investigation to determine the effect of light intensity on photosynthesis in an aquatic plant. So from here we know that what we are going to manipulate or change in an experiment is the what? Light intensity. So that becomes the independent variable. So what we'll now measure as a dependent variable is how photosynthesis takes place, perhaps the number of bubbles, okay, number of bubbles that will be given off, okay, per minute. Of course, we'll now set up the experiment. So let's start the investigation. Okay, let's do it like this. So let's start with the independent variable. Okay, now this is, so you say place an aquatic plant, since we are asked to use an aquatic plant, place an aquatic plant in a beaker submerged, okay, with water containing, okay, let's just say 20 grams of sodium hydrogen carbonate. Why did I start this way? Okay, for photosynthesis, there are several factors that can limit the rate of photosynthesis or affect photosynthesis. Light density is what we are looking at. Other things that can affect it is carbon dioxide concentration, amount of water available, temperature. These are things that can affect the rate of photosynthesis. So we have to keep all other factors constant. All right. So now let's go to the method now. Okay. Place a lamp, okay, at 3 distances, okay, from the beaker, okay, that is, let's say 10 cm cube, 10 cm distance, 20 centimeters and 30 centimeters respectively, okay, from the top. All right, and allow the setup for 2 minutes. You allow the setup for 2 minutes, okay, and that's the method you are going to adopt. So we'll place a lamp at 3 distances from the beaker, okay, 10 cm, 10 cm and 30 cm respectively from the top of the beaker, and allow the setup for what? 2 minutes. Okay, so let's look at our dependent variable. Now, how do we measure dependent variable and our result? Then observe and count the number of bubbles, okay, given off at the or during giving off during giving off during the 3rd minute, using, you now state the instrument using a stop clock. All right, that's the instrument to measure your result, and record the results. So that's what you do. You observe and record the result, okay, for 10 cm cube, 20 cm cube and 30 cm cube distances, okay, respectively. So that's how we'll take our dependent variable, the result, and the instrument. So once you do that, you now talk about your constant variable. What variables are you keeping the same to make your investigation reliable? Constant variable. We are going to use the same plant. We are going to use the same duration. Okay, of the experiment in each distances. All right, same amount of CO2, okay, or sodium hydrogen carbonate. All right, same temperature. Now all you need to do is, once you write this, you have to just pick about 3 or 4 of this constant variable. We are using the same plant. We are going to allow the experiment for the same duration. All right, we are going to allow it in the same room temperature, because these are factors that affect the rate of photosynthesis. 3 is enough, because each of these in the mark scheme will give you 3 marks. So you have 1 mark, 2 marks, 3 marks. For stating how you measure your result, that's another mark. The instrument you use is 1 mark. For outlining your independent variable, you have 1 mark. All right, the method, you have about 2 marks. So with this now, you have more than 6 marks already. So when you go on to reliability, what you do in terms of reliability, all you just say is what? Repeat the experiment, okay, twice, or you can say 3 times, and exclude. You say you exclude anomalous result or readings. What is the meaning of anomalous result? A result that does not conform to the pattern, that is wrong, that is not correct. We exclude it and we take your mean. So you exclude anomalous result. Then you come to safety. You can decide to add 1 safety precaution. Okay, safety precaution. Okay, look for 1 safety precaution that fits your experiment. Okay, and you get your answer. So that's what you need to do. So you just look for a safety or a precaution that you took during the investigation. Any relevant one will suffice or will do, and that's all about question 2. So like I told you, you have to follow the process of answering information. Take note of your independent variable, take note of the dependent variable, what you are going to measure. We are measuring the number of bubbles. You take note of the instrument you use to measure it, which is the stop clock. All right, and the variables you keep the same. All right, same amount of your source of carbon dioxide, which is sodium hydrogen carbonate, same temperature. All right, same type of plant or age of plant, duration of experiment will be the same. These are the essentials for answering paper. So we'll go on straight to the third question. The third question is on representing specimen. Here it's another important thing, a question that will give you the best, easy to answer. Just follow the prompt. It says this is a photograph of a cross section of a persimmon fruit, month fruit. Now what we'll be asked to do here is to draw. All right, drawing is one thing that is not difficult to do. The drawing usually is 4 marks. Now there are some essential things to take note of when you are asked to draw, and that's what I call the SOPD, all right, or SODP. the size of the diagram, okay, your outline size, the outline, the detail, and the proportion. Okay, I call it the SODP. All right, the size, the outline, the detail, and proportion. The size of your drawing, of course, must be bigger. Okay, the outline must be clear. Okay, single line, no broken line. Your drawing, the outline must have a single continuous line. All right, every detail in the diagram must be shown. Okay, major details must be shown, and it must be proportionate. All right, every detail, like in this diagram, have how many um internal circles containing disease. There are 6 seats, okay, and the other 2 compartments don't have seats, and of course they are central to the center of the diagram. Look at these projections here. There are some furrows at the top and at the side. So these are the details you should replicate in your drawing. Remember, your drawing must be bigger than the one you have given as the photograph. So let's try to draw this. Let me just do this so we can draw this. Let's see how we can replicate this diagram. Please, I'm drawing with a pen, so it might not be as clear. All right, so let's go with what we have. So it doesn't have, it's not art where you have to make it. So you go with continuous line that way, and you have that. Now this furrow here is not well done, so you do that. I'm using a pen on my tab, so it won't be easy, but if you have your pencil, you can always clean and erase, so you don't do broken lines. Broken lines like this will be penalized. All right, so you avoid this, you clean and do that correctly. Now we have the furrows on this side, this side, okay, and this side. Noted. The diagram is already bigger than this. All right, so we'll go back to the details in the diagram now. So we have 6 of this, that's one. We have another one, okay, that's 2. We have another one, that's 3. Then we have this one. So don't mind me, I'm trying to draw using this. That we have this, okay, we have this, and finally we have this. So that's what we have. Of course, this have fruit in it, fruit, 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 and fruit. Please, you have to avoid broken lines, and you don't shade. No shading, no painting. All right, you don't shade. So this shading, you just clean and redraw. No shading, no painting. Use a single continuous line, and that's what you have. So we are done with the drawing. With this, you have your 4 marks. So the outline, single clear line, we give you 1 mark. All right, your size must be bigger. Let's say this is 10. See, this diagram now is 10 or 100 millimeters. Let's assume this our diagram is 120 millimeters. All you just do is take your ruler, measure from this end to this end. All right, and you measure, you have 120, you get your magnification as times 1.2. so you also indicate your magnification in your drawing. All right, that this is what you've drawn is 100 millimeters when you measure from here to here, okay, depending on what you have in your diagram. And then you measure from what here and you have your magnification as 1.2. Very important, state your magnification. So you have the outline, we have the size, we have the detail, and the proportion in check. So that starts with how to draw. All right, so you have your 3 marks. It's not art, so it doesn't have to be so beautiful. Just make the outline clear and simple. Only important details are to be shown. Some questions will tell you shows this, show that. Don't draw individual cells, so follow the prompt in the question. Now they said measure line PQ on fig 3.1 Line PQ on 3.1 represents the diameter of the permission fruit and measure the length of PQ. So on measuring this length, since it's on the screen, it won't be exact, but on your paper, you just place your ruler and measure PQ in millimeters. So let's assume we've measured PQ and we have 100 millimeters. All right. Now, calculate the actual diameter of the permission fruit using the formula. So this formula is for calculation of magnification, the Maya formula, okay? Image size is I over A. Now that's a magnification is I over A. So you have the length PQ to be 100 over the actual diameter of the, okay, you calculate the actual diameter of the permission fruit over the actual diameter, and from the fruit, from what we have in the diagram, the magnification there is 2.2. All right, the magnification here is 2.2. So we are asked to find the actual size. So you just come in to have what 2.2. With A being the subject of formula, we have 100 over 2.2. Now they said the answer should be in what significant figure. So whatever you have must be to significant figure, three significant figure. Let's say you have 45.454 as the original answer. To three significant figure, we now have what 45 point, this is 54, so you have 44.5, and that's three significant figures. All right, now you don't need to repeat millimeters because they've already given you that here. These are the important things to note. Now coming to the B part, it says plants can make fat, starch and proteins. State the names of the reagents that can be used, okay, to test samples of plant tissue for starch and protein. Now for starch, we know we use iodine solution to test for starch. Iodine solution, original color is brown and it turns blue-black. For protein, we use burette reagent or burette solution, and it turns from blue to violet or purple without heating. Now it says describe the method you use to test a sample of plant tissue for fat. To test for fat, we can use the emulsion test, where we dissolve the sample of fat, okay, first in alcohol. Okay, the alcohol of course is ethanol. All right, then add what water and shake. All right, so when you add water and shake, the cloudy suspension is seen. Okay, that tells us is so. One thing is you dissolve, okay, because fat is soluble in ethanol, all right, but insoluble in water. That's how to test for fat. And final question here is,
This is what we do in the masterclass, really. We try as much as possible to ensure that we're able to properly, right, teach learners how they can properly understand, interpret the question as much as possible. We dive deeper than just a past paper to ensure that at the end of the day, they're able to fully understand any question that is being administered to them.
still on enzyme pectinase. It says enzyme pectinase is used to produce apple juice. Pectinase breaks down the cell walls in apple juice. This increases the volume of juice that can be extracted from the apple. Now, again, the essence of investigated the effect. They said the student investigated the effect of the concentration of pectinase on the volume of juice extracted from chopped apples. So they chopped the apples and added pectinase in different concentrations. So that's what the experiment is about. It says the masses of 7 samples of chopped apple were measured. All right. and the samples were put into different beakers. What is now changed here is the volume of pectinate solution added to the 6 beakers, all right, in different concentrations. So what was changed was what? Different concentrations of pectinase solution, which is an enzyme, but to use the same volume. All right, now they said the beakers were kept thermostatically controlled because temperature affects enzyme action. Now it says the content of each beaker were filtered through the filter paper, and the liquid was collected in a measuring cylinder. That's how much juice was obtained, was measured. So that's our dependent variable, the volume of liquid we obtained at each concentration. Okay, and that was recorded. So that's our result. Now they said state the independent variable in this investigation. What was changed? The concentration of what? The pectinase. So we change the concentration of what? Pectinase that was used. We changed the concentration. But what are the variables we kept constant? The volume used, the volume of pectinase, okay, was kept the same. Volume of pectinase used was kept the same. 50 cm cube was kept the same, and the temperature was also kept the same. They said was thermostatically controlled water bath. Temperature was kept the same. You can also pick every other thing that was kept the same, all right, in the experiment. And then they said it was left for 30 minutes, and duration, okay, duration of the experiment 30 minutes. All right, that's that with that. Now it says identify your hazard in the method described. What hazard and safety precaution do you expect? Since you are chopping the apples, you have to be careful you don't hold the apples on your hand to chop. You chop on a hard flat surface. Okay, so chopping the apples is a potential hazard. Chopping of apples is a potential hazard. And what do? You chop it on hard, okay, or flat surface. You have to chop it on a hard or flat surface away from your body, from the what? Body. All right, so you don't cut yourself. That's a precaution. Now it says the student did not repeat the investigation. All right, you only collected one set of results. There's no way it is better to collect several sets of results. You collect several results to identify. We are collecting several results to identify and exclude anomalous results. So that's why we collect several results, so we can have two or more readings and identify an anomalous result. An anomalous result is a result that does not conform to the pattern. This one says the 7th beaker of chopped apple did not contain pectinase solution. State what should have been added to this beaker to make it a control experiment. Experiments sometimes require a control, and since you are looking at the effect of pectinase enzyme, we know that temperature affects enzyme action, so you can put a denatured pectinase in, add a denatured pectinase, okay, so it won't have any effect on the apple, or you can just add normal ordinary water. That will also not have any effect on the amount of juice that you come up with. So one precaution is to say what? Add 50 cm cube of what? Denatured pectinase, or you can say boil. When you boil pectinase, of course, it's going to get denatured from high temperature. So denatured temperature, or you can say you add what? 50 cm cube of water. These are the possible answers you can pick. So anyone is good since it's just 1 mark. All right, and they say that is the result of the investigation. And you say a sample of 150 gram of chopped apples was placed in a beaker with the 0.8 pectinase solution. Use the information in the table to calculate the volume of liquid collected in the measuring cylinder. Now, these are the various concentrations of pectinase used. Now, what we need to do is said for this concentration, this value is per cm cube per gram, per cm cube per gram, meaning for 1 gram, meaning that 0.69 cm cube was collected for every 1 gram of chopped apple. So we are asked to look for how much volume will be collected for 150 gram SCM cube for 150 gram. Okay, that's what we need to do here. Now, so we say if 0.69 is collected for 1 gram, 150 gram, we collect what? What's it called now? XCM cube. So what we just do is say 150 times 0.69, and of course we get our answer to be 138 or so. Check, I think it's around 138, and we'll add the unit cm cube. So please take note of this. This is how to get answers. All right, the unit very important since the unit was not included here. Okay, so we have to include the unit. So every 150 gram of chopped apples will produce 138 cm cube of juice. That's what that means. Now plot a graph. Important thing about plotting a graph is that there are also things to note when we are to plot a graph. They said plot a line graph on the grid of the data in 3.1. So we have to plot a graph of this. We need to know what your independent variable is. So this percentage, what was changed in the experiment, or usually on the left, is your what? Independent variable. Okay, and what you have as your result, what you got out as your result, is your dependent variable. So whenever you are plotting a graph, you have to be able to identify what your independent variable is and what your dependent variable is. And I use this formula which I call the index formula, meaning that independent variable is always on the x-axis or the horizontal axis when you are plotting a graph. So coming to your graph, okay, we have our graph, and it's 4 marks. I'll tell you what the 4 marks usually entail. Number one, in drawing a graph, you have to label your axis. That's 1 mark. You have to use appropriate scale. You have to use appropriate scale. Then you locate your point. And draw a curve, a smooth curve. Okay, these are the things to do when you are making a graph. When you label your axis, you have to include unit with unit. All right, so let's do what we are talking about. Since we have seen the values on the table, we have the independent variable, percentage concentration of pectinase. All right, so that comes here. So that's concentration of pectinase. Concentration of pectinase. The unit is in what? Percentage. With this, you have 1 mark. Here is what? Volume of juice extracted. What is the unit in what? Cm cube. With this already, you have 1 mark without having to do anything. The next thing is use an appropriate scale, and scaling is very important. We have the lowest value from 0.2 and the highest value to 1.0. I have 1, 2, 3, 4, 5, 6 graduations. For the other one, you have between, okay, 0.6 to about 0.9. That's between 0.0 all the way to what, 1.0. So we go this way. Remember, this volume of liquid collected is the dependent variable. So you come here. So for concentration, since we are going between 0 and 10, you have 0.2, 0.4, 0.6, 0.8, and of course 1.0. That's on the concentration. Now on this side, you have the volume. We are going from 0.6 to 0.9. So we start with 0.1, 0.2, 0.3, 0.4, 0.5, 0.6, 0.7, 0.8, 0.9, and of course we have 1.0. Okay, you can decide to do 0.2, 0.4, 0.6, 0.8, and 1.0. Okay, still the same thing. I've just done this to make it easy for you to locate your point. So it means that in between this, we have 5 units between 0.1 and 0.2. So this is 0.12, 14, 16, 18, 0.2, 22, 24, 26, 28, and 3. So that's how to count the units in between. So let's come to our graph now. Our tables, we have at 0.2 we have 0.69. You come here at 0.2. Okay, let's even do it this way. 0.2, 0.4, 0.6. So 0.2, 0.69, 0.4, 0.77. So we have 0.69, 0.77. Now at 0.2, okay, let's go to 0.69. So 0.2 is here, 0.6 is here, 69 is just the last box in between. That's 0.69. All right, so that's in between there. And the other one is 0.4. We have 0.77. That's 0.72, 74, 76, 77 is just in between here. Then the other one is at 0.5. 0.5 we have 0.81, 0.6 we have 0.85. So at 0.5 we have 81 and 85. Okay, so let's do at 5. 5 is in between 4 and 6. We have 81. That's 8. 81 is just somewhere here in between, because it's not up to 2. And the other one is at 6. You have 85. So 82, 84, 85 is also in between here. Now, and the other two values is 0.6, you have 9292. So both at 0.8 and 1.0 is 0.92. To come to 0.8, we have 92, that's 92 and 92. What do we notice here? The value for 0.8 and 1.0 is the same. So you just take your ruler or you do a simple free sketch and you join all the lines together. You join the line, don't mind me, I'm using the hand and the drawing tool. So usually when you draw, you start from the first point. That's how to draw your curve. You start from the first curve. So we start from the first point on your graph and you stop your curve at the last point on your graph. Do not start from 0 if the unit doesn't start from 0. These are some details in getting your mark. So you start where it starts and stop at the last curve. All right, and you join the curve smoothly. You can use freehand or you use your ruler to connect one point to another. And of course, this here is showing that it's constant between 0.8 and 1.0. So with this, you have your 4 marks. So we've labeled our axis. We have 1 mark for that. We've used appropriate scale, another 1 mark. We've located our point, we have another 1 mark, and we have drawn a smooth curve, we have 1 mark. So that's 4 marks in a row. All right, so it's quite simple and easy to. They said describe the relationship between the volume of liquid collected and the chopped apple and the volume of liquid collected from the chopped apple and the concentration of pestilence. You are describing, that means describe the graph. What do you notice as the trend and the graph? We saw that the volume of liquid collected, okay, continued to increase as the concentration of pectinase was what increasing between 0.2 to 0.8. So you just put that down. You say volume of liquid, that's the juice now, juice collected was increasing as the concentration increased from 0.2% to 0.8%. That's first mark. It's a 2 mark question. What else do we see? Okay, from after, you can just say after 0.8%, the volume of liquid stayed constant. Stayed what? Constant. So that's 2 marks already. So you can describe your graph. You can say was increasing what? Steadily. The increase was steady until 0.8. That's 1 mark. After 0.8, no further increase. It was what? Stable. And that's all about paper 6 for this particular year, 2024, May, June, paper 6-1.
This is what we do in the masterclass, really. We try as much as possible to ensure that we're able to properly, right, teach learners how they can properly understand, interpret the question as much as possible. We dive deeper than just a past paper to ensure that at the end of the day, they're able to fully understand any question that is being administered to them.